All right, then. So in this lesson, we are going to talk about gameteogenesis, and it is the production of gametes in the gonads, right? So if it is the male gonad, it would be spermatogenesis, and is the formation of a sperm in the testis, right? And if it is oogenesis, is the formation of eggs or ova in the ovary, right? So the gametogenesis in males is known as spermatogenesis, and in females it is known as oogenesis, right? So let's talk about spermatogenesis in humans first. So here you're looking at a diploid parent cell, right? So sperm production takes place inside the tubules in the testis. So testis actually is actually the main organ, right? Where this sperm production will take place. So what happens is that, remember, the parent cell is always diploid, right? It's 2N. The diploid cell first will divide by mitosis to form a spermatogonia. So singular is known as spermatogonium, right? Plural is spermatogonia. So first, actually, by mitosis, the number divides, right? What happens after that? Now, again, if you're looking at this spermatogonia, all of them are diploid. All of them are 2N at this stage, right? They will then undergo growth and maturation. And what will happen then? They will then undergo the first division of meiosis, right? So, for example, here they have shown you one potential spermatogonia, which is undergoing meiosis. So, they will go undergo first division of meiosis and they will form two haploid cells, right? And as they form, as this diploid cell, actually, the one which actually this undergoes the first division of meiosis, it becomes the primary spermatocyte, right? So, the spermatogonium or spermatogonia, right? As they grow and they mature. So the mature form is known as primary spermatocyte. This primary spermatocyte is in the diploid form 2N. It undergoes meiosis, which is the first stage, right? And then it forms a secondary spermatocyte. So primary spermatocytes forms a secondary spermatocyte as it completes the first meiotic division, right? Then it undergoes a second meiotic division and then it forms four haploid spermatids, right? So spermatids are formed at the second meiotic division, right? And after that, these haploid spermatids, each of them will undergo maturation, and eventually they will form spermatozoa, right? So spermatozoon is a singular, or spermatozoa is plural. So this is the spermatogenesis in humans. Okay, so let me just repeat it one more time. So again, the parent cell is diploid 2N. So sperm cells, the production takes place in the testes. So first they will undergo mitosis. So as they undergo mitosis, the number will increase, right? So they will be known as a spermatogonia, right? Then they will grow and mature into a primary spermatocyte. Primary spermatocyte is still in the diploid state, right? 2N. Now this primary spermatocyte undergoes the first meiotic division and forms two haploid cells, which are known as secondary spermatocytes. The secondary spermatocytes then undergo the second meiotic division and they form four haploid spermatids. Then the spermatids will mature, right, into a spermatozoon, again, which is haploid, right? And like plural is known as a spermatozoa. So this is known as a spermatogenesis in humans. Okay, now let's talk about the process of oogenesis, gametogenesis in females. So the process of gametogenesis in females or oogenesis takes place in the ovaries, where these diploid cells are still at the 2N stage. They divide by mitosis, right? And they produce oogonia. So here you can see the plural is known as oogonia, the singular is known as oogonium, right? And what happens then after these, like these would grow and mature, right? And they would mature into primary follicles. So they would enlarge, right? They would mature, and then we call them as primary oocytes, right? So primary oocytes are still in the diploid stage, right? But what happens is that they would not complete the meiosis division. They would get stuck at prophase 1. So they would start meiosis, right? But they would get stuck at prophase 1 of meiosis 1, right? So generally what is happening is that time, like primary oocytes, as they're maturing, so what they do, they get encircled by single layer of follicle cells and they form primary follicles, right? Still at the birth stage. So the reason is that, like at birth, both ovaries actually contain around 400,000 primary follicles, right? So these primary oocytes, right, or primary follicles rather, they stay in prophase 1, in meiosis 1 throughout childhood, right? It means like they would stay dormant till the child has reached puberty, right? The moment puberty hits, these primary oocytes or primary follicles would now start to mature and differentiate, right? So the maturation and differentiation of primary follicle begins. So initially what happens, this meiosis 1 is completed because it was stuck at prophase 1, right? So this gets completed. And what is happening at this stage is that one of the cells actually, during cytokinesis, actually what is happening, there is uneven distribution of cytoplasm. So one of the cells actually gets converted into a secondary oocyte, so it gets most of the cytoplasm, while the other converts into a polar body, right? So this is known as the first polar body. So this degenerates, right? So this secondary oocyte will actually then undergo maturation after puberty. All right then. Now, so as I was saying, cytokinesis evenly takes place, right? So we have this large secondary oocyte, which is now haploid, right? Because the first meiotic division is completed, right? And we have this small polar body, which is known as the first polar body. Now what will happen, the second meiotic division will take place only if this secondary oocyte actually is stimulated by a sperm cell. So what is happening, the secondary oocyte, right? Like it will mature then. And then what will happen, like it will stay a secondary oocyte and it will be released once a month. But it will complete the second meiotic division only if it is stimulated by a sperm cell, right? So at this moment, what happens, it would complete the meiotic division, right? Again, the same thing would happen. There will be uneven distribution of cytoplasm. So one of them will develop into an ovum, right, after maturation, and this will be happening after ovulation, and the other would develop into a polar body, which will definitely degenerate. Now, this process, as we are saying, is takes place once this secondary oocyte is stimulated by sperm cell. So what happens is sperm actually, as it fuses with the female, the ovum, right? So the meiotic division is completed after that, after the fusion, right? So this mature ovum, the lifespan is very, very short and is subjected to fertilization, right? Because this second meiotic division will not complete. The gametes will not like form till the time it is fertilized, and then it becomes a diploid heterozygote, right? So for this to understand, actually, I want you to have a look at a video about fertilization, and then I hope this process will become more clear to you.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads called microtubules pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
All right then, so let's talk about gametogenesis in flowering plants. So the male gametes are produced in the anthers and female gametes are produced in the ovules, right? So when we talk about the male reproductive organs in plants, right, it's known as the stamen. So stamen is the main reproductive organ that is found there in the plants. So it is made up of two parts. One is known as anther and the other is filament, right? Filament provides the support while these gametes are actually produced in the anther. So here you can look at the structure. So this part here that you're looking at, this stamen, and it consists of anther and filament, right? Anther is the part that produces the male gametes. So here you're looking at another structure, right? So filaments, actually, the filaments that you're looking at here, they support the anthers, right? And the pollen grains are produced in the anthers. So pollen grains are the male gametes found in flowering plants. So filaments, besides producing or giving support, basically, they also contain the vascular tissue, right, which supports the or transports the food materials required for the formation of pollen grains, right? So those vascular tissue, right, it's found there in the filaments as well. Now, anthers are usually made up of 4 pollen sacs, which are arranged in 2 pairs. So if you look at this diagram here, so you're looking at these 4 pollen sacs, 1, 2, 3, and 4, right? And they're arranged in two pairs. So this is how they are arranged. So this about the anther and filament providing support to it. Okay, so let's talk about the development of pollen grains in the anthers. So in the anthers, again, we have these diploid cells, which we call as pollen mother cell, right? So it's 2N. And as they divide by meiosis, they give rise to 4 haploid cells, right, which is N. So this is a pollen mother cell diploid, and it undergoes meiosis, and we have these 4 haploid cells now. Now, the thing is, these cells will now undergo mitosis, right? So what happens is that the cell itself does not undergo cytokinesis, but the nucleus divides, right? So nuclei of each of the haploid cell divides by mitosis, but the cell does not divide. It means cytokinesis is not taking place. But what happens is that the cells that we are actually getting in this case is that each cell would have a haploid nuclei, right? These cells then mature into pollen grains. These are the mature pollen grains now, right? And as they mature, they are surrounded by protective wall, which is made up of tough exine and a thinner intine. So we look about that. So this is here what they've shown you, right? So we look at the detailed view in the next picture as well. And one of these haploid nuclei, if you're looking at these two, one of them is known as a tube nucleus and the other is known as generative nucleus, right? So these are the male gametes, pollen grains, right? Individually, like they develop into pollen grains, we're looking at these, right? But they start with a pollen mother cell, which is 2N. They undergo meiosis, meiosis 1, meiosis 2. They have these 4 haploid cells, which then undergo mitosis, but the nuclei divide, right? But the cell itself does not divide. So each cell has a haploid nuclei now, and each of them then matures into a pollen grain, right? And these nuclei, one of them is known as the tube nucleus, and the other is known as generative nucleus, right? So here's another diagram of that. So you have this mother cell. It undergoes meiosis 1, so you have 2 cells, right? And it then undergoes meiosis 2, so we have these 4 haploid cells there, right? And then these pollen grains, again, they separate. And then what happens? Now in this stage, as they're undergoing mitosis, the cytokinesis would not happen, but the nuclear division would take place. So nuclei would divide. And these are the mature pollen grains, right? So as they develop, they have this outer membrane, which is known as exine. So it's quite tough, right? While the inner membrane, jo intine hai, it's smooth. So you can see as well this light orange layer that you're looking at. So it is smooth, and the outer membrane exine is tough, right? They can you can look at the generative nucleus, right? And you can look at the pollen tube nucleus, right? So this is how these are the male gametes or the pollen grains. All right, then let's talk about the female reproductive organs now. Right. So in this case, in plants, it is known as the carpel, and the carpel itself consists of stigma, style, and ovary. So these 3 components actually make up the carpel, right? So stigma is a surface on which the pollen lands, and the pollen tube grows down to the ovary, right? So this part here you're looking at is the stigma. To this part, the pollen will land, so that you can see the yellow dots there. So pollen land on the stigma, and then they travel down, right? So, so what happens? This pollen grain actually it develops a pollen tube, and then it goes down, right, into the ovary. Then we have this part which is known as the style, right? The style connects the stigma. So this is the stigma to the ovary. So this part here that you're looking at is the ovary, and ovary contains the ovules. So here you're looking at the ovules, right? So ovules are the female gametes, and these contain a single egg nuclei, right? So this is the female reproductive part of the flowering plant. So this is carpel, consisting of stigma, style, and ovary. All right, then here's another picture. So you can look at this part here, the stigma, this is the style, ovary, and then you can see like this is the ovule here, right? All right, then. So how the embryo sac develops here. So inside each ovule, right, again, we have a large diploid spore mother cell. So this is a spore mother cell, right, diploid 2N, right? It undergoes meiosis, and we have these haploid 4 cells then, right? What happens then? These cells would now undergo mitosis. But what happens is that as these cells, before they undergo mitosis, all of these 4 में, ठीक है, 3 of these degenerate, right? But only one of these actually becomes the surviving haploid cell, right? So there were 4 cells initially haploid, but 3 of them would degenerate as mitosis starts, right? So 3 of them would degenerate, okay? But the 4th one would actually undergo mitosis, and we have now haploid cell which has 2 nuclei. Again, cytokinesis does not occur, right? But we have 2 haploid nuclei in the cell, right? So this forms the embryo sac. And the 3 cells here, with this part here is not the micropyle, they are crushed, they are degenerated, right? So this is the one surviving haploid cell. And this haploid cell actually then develops into an embryo sac. All right, then. This embryo now grows and gets matured, right? And you can see that mitosis keeps on continuing. So these were 2 haploid nuclei. So they undergo division. So again, now from 2, we have 4 in the second division. This will undergo another mitotic division, and now we have 8, right? So embryo sac grows larger, and you can see this is the embryo sac. These are the 3 that get degenerated. So you can have a look at here also. And by the time this third mitotic division takes place, they're completely, these 3 have completely, they're not there anymore, right? But you look at the embryo sac. Embryo sac, it keeps on getting bigger, right? It divides. And then embryo sac gets larger. Its haploid nucleus actually dividing by mitosis, but it divides 3 times. So see, from 2, it becomes 4, and now it's 8 now. Right. So what happens is that these two nuclei, the one in the middle, actually they fuse together and they make one nucleus, right? And the one which is here, because 3 move here, out of 8, 3 move here, 3 move here, so 6 and 2 are here. So the 2 in the middle fuse together, right? But this one here in the middle, this is the one which actually develops into a female gamete. This one here, right? So the pollen grain which actually fuse with this one at the time of fertilization. Right, so this is how this embryo sac develops in the female reproductive parts of the plants. Right, so it starts with 2N diploid cell, the diploid spore mother cell. They undergo meiosis. We have 4 haploid cells. 3 of them degenerate. Only 1 develops into an embryo sac. It then undergoes mitosis. So these haploid nuclei, they undergo mitosis. So we have in the first division we have 2 of these. In the second we have 4. In the third division we have 8. So out of these 8, the 2 in the middle, they fuse together. And the one at the top here in the middle, it is the one which develops into the female gamete. And this is then fertilized by the pollen grain, right? But now the thing is that in plants, unlike animals, gametes are not formed directly by meiosis. Because as we just look at gametogenesis in humans as well, so they were being formed by meiosis. But in plants, gametes are not formed directly by meiosis, right? Meiosis takes place, but it is used in the production of pollen grains and embryo sac, right? So pollen grains and embryo sacs are produced there, right, by meiosis, but are then formed inside these structures by mitotic divisions, right? So meiosis use जरूर हो रही है pollen grains और embryo sac बनाने में, but then gametes जब बन रहे हैं, ठीक है, they are formed inside these structures by mitotic division. So this is one difference between gametogenesis in humans that we just studied a while back and gametogenesis in flowering plants.